Panama, June 1905. The Canal Zone records 62 new cases of yellow fever in 30 days. By August, the count is 27. By December of the same year, the count is 1. The man who shrank that number did not invent a chemical, did not patent a device, and did not bill anyone for a service. He drained ponds, oiled gutters, and screened porches. For pennies per acre, he wrote the entire method down. 120 years later, the industry that grew up to fight mosquitoes quietly stopped recommending most of what he wrote. 1. The man who drained Panama, William Crawford Gorgas, was born in Mobile, Alabama, in October 1854. He failed the entrance exam to West Point 3 times. He went to medical school instead, Bellevue Hospital in New York, graduated 1879, because the Army would still take him on as a surgeon. In 1882, posted to Fort Brown on the Texas border, he caught yellow fever during an outbreak that killed most of the small garrison around him. He survived. The nurse who sat with him through the worst 3 weeks of his life was named Marie Duty. She had been infected by her own sister, who died. She survived as well. Gorgas married her 3 years later, in 1885. He came out of that fever ward with 2 facts pressed into him. The first, yellow fever was something physical and transmitted, something a person could be cured of by surviving, not a punishment of climate or filth. The second, he was now immune. The Army could send him into the worst disease zones on the planet, and he would walk out alive. In 1898, after the Spanish-American War, the Army sent him to Havana. He arrived as chief sanitary officer of a city where 784 people had died of yellow fever the previous calendar year. He left it 4 years later with the case count at 0. How he did that is the story almost nobody hears in full. 2. Carlos Finlay and the 19-year silent 17 years before Gorgas arrived in Havana. A Cuban physician named Carlos Finlay stood at the International Sanitary Conference in Washington and proposed that yellow fever was carried by a mosquito. The year was 1881. He named the species, a small domestic mosquito he had observed breeding in household water jars. He described the transmission cycle in unusual detail. The room laughed. The senior delegations went home and continued treating yellow fever as a disease of bad air, dirty linen, and contaminated cargo. For 19 years, Finlay's paper sat in a Cuban medical journal that the European medical establishment did not read. He kept performing experiments. He let infected mosquitoes bite consenting volunteers. He logged incubation periods of 3 to 6 days. He published in Spanish and in French. The world, as a structure, was not arranged to receive his findings. There was no institution that would lose money if he was wrong, and no institution that would gain anything obvious if he was right. Finlay was nominated for the Nobel Prize 7 times before he died in 1915. He never received it. In 1900, the U.S. Army Yellow Fever Commission, led by Walter Reed, finally went to Cuba to test the theory. They tested it the only way it could be tested in 1900. Human volunteers, controlled infection, observed outcomes. Jesse Lazier, one of the commission members, allowed himself to be bitten. He died. The theory was confirmed. Gorgas read the commission report in early 1901. He did not run a chemical campaign in Havana. He ran a mapping campaign. Every cistern, every barrel, every cemetery vase, every cracked roof gutter holding a thumb's depth of standing water, catalogued, drained, oiled, or screened, house by house, block by block. Cases collapsed within a single fever season. In March 1904, the Army sent him to Panama. The French had abandoned their own attempt at the canal in 1889, after roughly 20,000 workers died on the project, most of them to mosquito-borne disease. The American Canal Commission gave Gorgas command of sanitation. Theodore Roosevelt backed him personally after some early political resistance. Gorgas organized 4,000 workers into what he called mosquito brigades. Across roughly 500 square miles of jungle and town, the brigades poured 600,000 gallons of oil over standing water, applied 300 tons of sulfur in fumigation, and screened every habitable structure with copper mesh. By November 1906, the Canal Zone recorded no further deaths from yellow fever. Not one, for the remaining 7 years of construction. The canal opened in August 1914. Gorgas became Surgeon General of the U.S. Army the following year. He published his methods openly. His 1915 book, Sanitation in Panama, is still in print. The library copies have not been pulled. The book is still in print. The habit is not. 3. Inside the mosquito. A life lived underwater. The reason the method worked is biological geometry. A mosquito's life has 4 stages: egg, larva, pupa, adult, and 3 of those 4 happen entirely underwater. A female Aedes aegypti lays between 100 and 300 eggs per batch, and she lays them only where water sits still for several days. 48 hours later, the eggs hatch into larvae. The larvae hang at the surface and breathe through a small tube called a siphon, drawing air through the water's surface tension. 7 to 14 days later, they pupate. 2 days after that, they fly. Break any of the 3 aquatic stages and the next adult never lifts off. Cervix tension itself is the leverage point. Clean water at room temperature holds a tension of roughly 72 millinewtons per meter, enough for a larval siphon to anchor on the underside of the meniscus. Add a single drop of ordinary dish soap or vegetable oil to a still container, and the tension collapses to around 30. The larvae cannot hold the surface. They sink and suffocate within minutes. Eggs deposited afterward fail to develop. For larger water, ornamental ponds, rain barrels, livestock troughs, water meant to be kept clean, there is Bacillus thuringiensis subspecies israelensis, abbreviated BTI. It is a soil bacterium that produces 4 specific protein crystals: Cry4A, Cry4Ba, Cry1Ma, and Seat1A. Larvae filter feed the crystals into their gut. The alkaline midgut of a mosquito larva, at a pH between 9 and 10.5, dissolves the crystals into active toxins. The toxins bind to receptors that exist only on the gut walls of Aedes, Anopheles, and Culex larvae, and the cells rupture within hours. Humans, pets, bees, fish, frogs, songbirds, all have acidic stomachs and do not carry those receptors. BTI walks past them untouched. Sold as floating tablets called mosquito dunks, each one treats roughly 100 square feet of standing water for about 30 days. The retail price runs about 50 cents to $1 per tablet. The chemistry is selective. The cost is small enough that the cost itself is almost the point. 4. How the method was quietly forgotten. The sanitation maps Gorgas drew in Havana still exist in the U.S. National Library of Medicine. The protocols he wrote up in Panama in 1914 were taught in U.S. public health schools through at least the 1950s. Then something quiet happened. Housing built after about 1980 began to include surfaces that hold water without anyone noticing. Gutters with insufficient pitch, air conditioner condensate trays that nobody empties, corrugated drainage pipes that pool at every elbow, decorative planters with hidden saucers underneath, irrigation backflow valves, recycling bins with ridged interiors. The method was not refuted. The geometry of where it would be applied simply changed faster than the habit of looking. If a grandmother somewhere in the family line kept rain barrels covered with cloth, or turned buckets upside down before going indoors at sundown, or flicked the saucers under her potted plants out into the dirt every evening without quite explaining why, she was reading a paragraph from a book her own grandmother had memorized. The reason had a name. The reason had a Cuban doctor, an Alabama Army surgeon, and a Panama jungle behind it. Nobody handed her the citation. She did the work anyway. The chain ran on habit, not on credentials. 5. The economics of a method that cannot be sold. The U.S. residential mosquito control service market generates an estimated $2 to $3 billion in annual revenue. A standard treatment from a national franchise. Mosquito Joe, Mosquisquad, Mosquito Authority, and the regional equivalents runs between $80 and $150 per visit. A full seasonal contract runs $350 to $1,000. The chemistry applied is almost always a synthetic pyrethroid, an adulticide that kills mosquitoes physically present on the day of spraying and degrades under sun and rain within 2 to 3 weeks. The next generation, already underwater in a forgotten saucer, 6 feet from where the spray truck parked, hatches on schedule. There is no patent on a drained gutter. There is no recurring revenue model on a tipped saucer. There is no marketing budget for a 50-cent BTI tablet. The method Gorgas published openly in 1915 cannot be owned, billed, or scheduled, and an industry built on quarterly contracts has no economic reason, structurally, to point the customer toward the bird bath. This is not a conspiracy. It is the shape of who pays for what. A method that works once and stays done does not pay anyone twice. 6. Sayaxché 2015. 18,000 eggs per month. The most recent large-scale field test of the source reduction principle ran for 10 months in 2015 in the town of Sayaxché in northern Guatemala. Population roughly 15,000. Climate hot and humid. Dengue endemic. A research team led by Gerardo Ulibarri of Laurentian University in Ontario, working with the Guatemalan Ministry of Health and the National Institute of Public Health of Mexico, built a low-cost trap they called the Ovalanta. 2 50-centimeter sections of an old car tire, fitted into a mouth shape, with a drainage valve at the bottom and a strip of Pellin paper floating on water inside. The materials cost less than $4 per trap. 84 Ovalantas were deployed across 7 neighborhoods. Female mosquitoes, drawn by the dark cavity and the warm, sheltered water, laid eggs on the paper strips. Every 2 weeks, the team drained the water through fine fabric, collected the paper, and destroyed the eggs with fire or alcohol. The drained water was poured back in. The water contained an oviposition pheromone released by every female that had visited the site. The more females visited, the stronger the chemical signal became to the next females. The trap got more attractive the longer it ran. Over the 10 months, the Ovalantas captured roughly 18,100 eggs per month. The control group, using standard ova traps, captured about 2,700. Roughly 7 times the catch, at a fraction of the cost of either larvicide or adult spraying. About 1/3 the cost of larval programs. About 1/5 the cost of pyrethroid fogging. The same paper noted, as a secondary observation, that no new dengue cases were reported originating in the study zone during the trial period in a community that would normally expect 2 or 3 dozen. The authors flagged that figure carefully as tantalizing but anecdotal. It has not been replicated at a controlled scale large enough to be called proof. $4 per tire, 18,000 eggs. 1 Guatemalan dry season. The arithmetic in the title is not a slogan. 1 female mosquito lays 100 to 300 eggs per batch and produces, under favorable temperatures, between 5 and 7 batches across a roughly 30-day adult life, somewhere between 500 and 2,100 offspring per individual female. The second generation, if it reaches maturity, repeats the same fertility on a larger base. Eliminate 1 persistent standing water source on 1 suburban property, and the prevented adult emergences across a full breeding season run into the low tens of thousands. 20 houses on the same residential block, working the same audit on the same fortnightly cycle, prevent emergence into the hundreds of thousands. A town the size of Sayaxché, 15,000 people, perhaps 4,000 households, clears the math past 1 million within a single breeding cycle. A mid-size county clears it past 10 million by midsummer. The number in the title is not hyperbole. It is what the reproductive arithmetic does once the saucer goes empty. Seven. The home protocol. Audit, cut, treat, trap the full protocol, in the order it should be done. First, the property audit. Walk the perimeter slowly once. Look for anything that holds water for longer than 3 days. The usual list. Roof gutters, downspout extensions, AC condensate trays, corrugated drainage pipes, water pools at every joint, pot saucers, bird baths, cemetery vases, kiddie pools, kayaks stored upright, tire piles, tarps with low spots, plastic toy buckets, wheelbarrow basins, clogged storm drains, tree hole cavities, irrigation valve boxes, and the ridges inside outdoor recycling bins. Every container either empties, inverts, drills a drain hole, or receives a BTI tablet. Audit on a 14-day cycle, because 14 days is the maximum egg-to-adult window for most domestic species. Second, the resting harborage cut. Adult mosquitoes spend the daytime hours resting in shaded, humid vegetation. Dense, low shrubs against foundation walls, ivy curtains, tall grass under eaves, the underside of deck planters, woodpile crevices. A modest trim, opening these refuges to airflow and direct sunlight, removes most of the daytime hiding layer. Mosquitoes are weak flyers and dislike dry, turbulent air. Cutting back is half the protocol that nobody mentions in spray truck advertising. Third, BTI in any water that cannot be drained. Ornamental ponds with fish are safe. Rain barrels meant for garden use are safe. Livestock troughs are safe. Drop 1 floating tablet per 100 square feet of water surface area. Replace every 30 days through the active season. The tablets dissolve slowly. The bacterium continues to produce its protein crystals as long as the material remains in suspension. Fourth, a single oscillating fan on any porch or patio meant for evening human use. Adult Aedes mosquitoes cruise at roughly 1 to 1.5 miles per hour. A standard pedestal fan on its medium setting produces airflow several times that speed. The fan does 2 things at once: physical displacement and dispersal of the carbon dioxide plume from human breath, which is the long-range chemical signal female mosquitoes use to find a host. Carbon dioxide as a mosquito attractant was first reported by Rudolphs in 1922 and quantified by W.C. Reeves at the University of California in 1953. The receptors involved sit on the maxillary palps and detect concentration gradients of as little as a few hundred parts per million above background. Scatter the gradient and the chemistry that brings the female toward the host stops working. The fan is not symbolic. It is interrupting a signal. Fifth, for properties with persistent pressure, the home version of the Ovolanta, a dark bucket, opaque plastic, or wrapped in black, holds about a gallon of water. A strip of cardboard or rough paper floats on top. Refill from the same water after each cycle. Do not discard the pheromone-loaded liquid. That signal is the entire point. Strain and incinerate the paper every 10 to 14 days. As an attractant assist, a small sealed bottle of fermenting sugar water, 1 cup warm water, a 1/4 cup of brown sugar, 1 gram of dry baker's yeast, can be placed near the laying bucket and vented through a short tube. The yeast produces a steady, low stream of carbon dioxide for roughly 2 weeks as it converts the sugar through the reaction. C6H12O6 yields 2 ethanol and 2 carbon dioxide. That CO2 plume pulls gravid females toward the dark water, where they lay eggs that the operator then destroys. The yeast bottle on its own, without the laying trap, does not kill mosquitoes in meaningful numbers. It's matrax. The kill happens in the bucket and at the paper strip. That is the whole system. Audit, cut, BTI, fan, bucket. What the protocol does not do. It does not protect against mosquitoes flying in from a neighboring property that is not running the same audit. The effective working radius of one fully cleaned property is roughly 100 to 150 meters under still air, less in wind. It does not work in a flood week, because new standing water appears across the landscape faster than any audit cycle can catch. BTI loses potency in direct sunlight and must be replaced on schedule rather than left alone. The dish soap surface tension method is for sealed containers only. Do not pour soap into a fish pond or amphibian habitat, as the surfactant will harm anything else that depends on the water. And the entire approach is preventive, not reactive. It will not clear an existing adult swarm overnight. It clears the next generation and the one after that. The first measurable drop typically appears in week 3 or 4. The structural drop appears in month 2. Closing. William Gorgas drained ponds in 1904 because he had no other option. The synthetic chemistry came later. The trucks came later. The quarterly contracts came much later. 120 years on, the saucer under the tomato plant on the back step still works exactly the way it did the day he wrote the protocol down. Tip it out tonight. Walk the perimeter once. Count what holds water. In 2 weeks, come back to this video and post what was found, what was emptied, and what the count was the second time around. The comment section below is the field study now. The next person who reads it is the next Mosquito Brigade.